Okay guys, so today we're going to start chapter 13, and this is going to be a chapter all on DNA. We're going to talk about the history behind DNA, what DNA is made out of, what DNA pairs with, how those pairings happen, and then we're also going to talk about how we learned that DNA was our inheritance material, our heritable material. So back in the 1950s, two scientists, James Watson and Francis Crick, they were the first ones to discover, that DNA is in a helical form. In other words, it's not going to be a straight line. And we know DNA is made of a double helix. Well, there's a bunch of different ways a double or two strands of DNA could go. They could go straight, or they could spiral like a staircase. And James Watson and Francis Crick were the scientists who said that DNA is actually a helical form. So every single one of your cells has DNA. Every single cell for humans has 46 chromosomes for most people. Now, every time those cells reproduce, all of our new cells also have 46 chromosomes. So if we start off with one cell, guys, if we start off with one cell, and that one cell is, I'll just draw it here on the bottom. If that cell right there has 46 chromosomes, and if we were to go through cell division and make two new cells here, so one there and one there, if that first one has 46 chromosomes, that's our number we want, obviously. But if we divide it in between the two cells down here, well, if you take 46 and divide it by 2, that would leave 23. So that's not going to work, because we want all of our body cells to make sure that they have 46 chromosomes and not 23. So if this cell has 46, and we want these to have 46, something has to happen up here in order to make sure that we can keep that number the same. We can conserve that 46 number of chromosomes. So what we do is right before our cells are going to reproduce, and we learned this already in mitosis, is we are going to make a copy of our DNA, bringing our number up to 92. And then that 92 for chromosomes can be split in between those two cells. All right. So here is James Watson and Francis Crick. James Watson on the left there is still alive. Francis Crick is no longer with us. And here was their original structure of DNA. And you guys can see it starts to spiral like a staircase around this metal rod in the middle. All right. So for DNA, guys, and we kind of already went over this picture, but DNA is made out of two things. We have the DNA portion of it, and then we have this protein called histones. And these histones are going to pretty much keep the DNA in a shape, in a certain shape that we want that DNA to be in. Whether it's chromatin during interphase when the cell is not dividing, or if it is chromosomes when the cell is dividing, we are going to have this DNA wrapped around these proteins called histones. And all that's going to do, guys, is keep our DNA kind of organized, keep it in one area, and keep it condensed, because DNA would be extremely long if we didn't have it coiled around all these histones. So for DNA, we're going to start to go over how we determined this was our molecule of heredity. But how we first discovered our DNA was we were studying bacteria and some viruses. So anytime you guys, when we think about viruses, the goal of a virus is to infect the bacteria, or we could call it a host cell, and get that cell to reproduce. And when that cell reproduces, it's no longer reproducing the cell, it's actually reproducing little baby viruses. And what we call that is a bacteriophage. Okay, so that's a bacteria cell that is going to be infected by a virus, and it's pretty much like a virus making machine. So this was now just in the time period. This was before the 1950s. This is around 1930s. So this was before James Watson and Francis Crick discovered that DNA is in this helix form. But also during this time, we didn't really know that DNA was this hereditary material for our bodies. So they were doing different experiments, and they used two different strains of bacteria. They used an S strain, which is bad, and they used an R strain, which is not bad, okay? So here's what they did. Again, S strain bad, R strain not bad. Now, the first one, they injected a mouse with the bad strain, the S. And obviously, just as expected, the mouse dies, okay? So, mouse died. And now, since the S1 is bad, or virulent is another word for it, I'll write that here. Virulent. And that's just a bad strain. If we inject these bad bacteria cells into the mouse, it should die because the S strain is bad. Now, the second one, R strain, we said was harmless. So if we inject the mouse into that, nothing's going to happen. The mouse should survive, which mouse healthy, okay? The next one is, and if you guys have ever like boiled water or anything like that, if your parents ever had, another child, you might see them boiling bottles and stuff like that to just try to kill all the bacteria in them. But what happens, guys, is when we heat bacteria up, we boil bacteria pretty much, it dies. So what they did in the third one is they heat killed the S strain. So they heat killed the bad strain. And what they found was after they heated up, now it'd be to a very high temperature, if they injected that one where the S strain got heat killed into the mouse, mouse was fine. No problem there. But the last one is the weird one. So what they did was they took these heat-killed S cells. So the heat-killed S cells were harmless. And they also took living R cells, which we know. So living R cells right there, the mouse was fine at the end. Heat-killed S cells, the mouse was fine at the end. Mix them together, and the mouse dies. And this was where the whole experiment, this was like the basis for it, and it was like, okay, why did this happen? The R cells didn't kill the mouse. The heat-killed S cells didn't kill the mouse. Put them together and the mouse dies. Why? So here's what they found out, was when they mixed the heat-killed S cells with the living R cells, the R cells actually went through a DNA transformation. And what happened was that foreign DNA from the S strain that was dead, but the DNA is still there, it went into the R strain. And it transformed that DNA to make the harmless R strain into, so it made the harmless R strain into S strain. And I'm not talking about heat kills the S strain anymore, because remember the R was living in that last one, right? You can see here it was living. So it transformed it into regular S strain. And we know that regular S strain, that was the first one there, the mouse died, okay? So that's what a transformation is. They were taking the DNA and putting it into another bacteria, the R1, and it transformed the R bacteria. So second one here, second experiment, was by Oswald Avery. And what they did in their experiment here, they identified the transforming substance in that transformation. So they kind of took that previous work with the R strain and the S strain, and they said, okay, well, what's causing the mouse to die? Is it DNA? Is it RNA? Is it a protein? Is it something completely different? They came to the conclusion that it was DNA, because before this, guys, we didn't know. We did not know that DNA was so important to us. It was all kind of new information. But anyway, so I talked about bacteriophages already, and we'll go over the experiment here in a bit. A bacteriophage, just to review, guys, is simply a bacteria that gets infected by the virus, and it becomes a mini virus making machine. All right, now all a virus is, guys, and I'm going to draw a bacteriophage virus here. Okay, all it is, this is called a protein coat surrounding it. All right. All right. So we got a protein coat surrounding it. We have some DNA in here. So there is your DNA. And there is a little tiny tube coming downward. And what that does is it allows this DNA to latch on to a bacteria cell and inject this DNA into it to then transform the bacteria DNA so that the bacteria DNA will make little baby viruses, okay? But anyway, that's pretty much all a virus is, guys. It's DNA, or like the coronavirus, a new one, that's an RNA virus. So there are some RNA viruses out there. But all it is some kind of genetic material enclosed in a protein. So viruses really aren't even considered living because they don't meet all the needs of living things, the characteristics of life. They don't have all of them. All it is DNA in a protein. All right, so we already talked about this one. Viruses, they'll latch onto the cell membrane of a bacteria, and then they will take over that cell, okay? And the goal is just to make other baby viruses. So here is the bacteria cell down here in this brown kind of orange color. There is the virus up top. And you guys can see that there is that little tiny tube there, and that is the DNA going from the protein up here into that bacteria cell. And like I said, it'll go to that DNA and transform the bacterial DNA so that it becomes a baby virus producing machine. So here's the experiment that went along with all this, the bacteriophages. So Hershey and Chase. Once again, they kind of took Avery's experiment and explained it in a little bit more detail and did so through all their conclusions on their experiment. So what they did was they determined that DNA was the genetic material. And they used the bacteriophage to do this. So let me draw some things out for you here. Okay. So what they did, and I'm going to draw my bacteria cell here. All right. And I'm going to draw the virus attaching to the bacteria to the best of my ability here, which is not great. Okay. So now what I'm going to do is I'm going to draw the protein in red, and I will draw the DNA inside of that protein. Just make it blue. Okay. All right. So, blue, that's our DNA. Red, that is our protein. So what they did was they fluorescently dyed parts of the bacteriophage here, or I'm sorry, the virus, and they dyed the protein with a different isotope than the DNA. So what they did here, guys, is they used two different isotopes. They used sulfur, and they also used phosphorus. So for the protein, they used radioactive sulfur. For the DNA, they used radioactive phosphorus. Could also say phosphate, but phosphorus isn't phosphate. So why did they use sulfur and why did they use phosphorus? Well, They use these two because proteins only have sulfur. If we go back to the macromolecule chapter, proteins only have sulfur and they do not have phosphorus, okay, in the monomer for it, in the amino acid. DNA only has phosphorus and it does not have sulfur in the monomer of DNA, which is a nucleotide, okay? So proteins, guys, if you remember, we have like, if we want to go over the monomer of it, we had a central carbon, we had an R group on top, we had a hydrogen down below, we had an amino group on one side and a carboxyl group on the other side. But what they did was that in that R group is there was sulfur in it. DNA, if you remember our monomer of a nucleotide, we had whatever our base is, A, T, G, or C. We had our deoxyribose sugar, and then we had a phosphate group, and that phosphate group is where the phosphorus is going to be. So they let the virus infect the bacteria. And I'm going to draw the bacterial DNA right here. All right, so there is our bacterial DNA. All right. So after this, they let the virus do its job. Okay. The bacteria then started to produce these little baby viruses. And what they did was they removed the bacteria, okay, from this thing. And how they did this is they used kind of like a blender, honestly. It's like a scientific blender. And they blended all these pieces up. So it separated the viruses from the bacteria. And now we have this smoothie, I guess you can call it, full of bacteria and viruses. Then what they did was they put it in something called a centrifuge. And a centrifuge is pretty much, if you ever went to like any kind of amusement park, you ever see one of those wheels that just spins like this, and you're like on the outside, like, oh my goodness, I can't move. But you get spun around kind of like a, I don't know, like a top or something like that. But anyway, so what they do with the centrifuge, guys, it's a machine that spins at very, very fast speeds. And you have little tiny test tubes in it. And what it will do is it will separate the contents of the test tube based on the densities. So here's our test tube. The low density objects will go to the top. The high density parts will go toward the bottom of our test tube. And this is actually what the machine they use whenever they're separating the blood from blood plasma. So they will actually use this machine, and the plasma, the liquid watery portion of your blood, will separate from the red blood cells. And you'll actually have a clear layer and a red layer, which is kind of cool. So like I said, guys, they put this stuff with the bacteria, the viruses, they put it in a blender. They hit frappe, and the bacteria is separated from the viruses. Then they put it in a centrifuge, causing it to spin. And what happened here was all the viruses, let me not use that red, but all the viruses have a lower density, so the viruses were on top. And then down below here, at high density, we had our bacteria cells. Okay? Now, the viruses up here, we're going to see both the sulfur and the phosphorus in our radioactive protein and DNA. We're going to see that up there. Okay? But, come on, lights. But we don't care about what's up top, because we don't care about the viruses. We know the viruses had the protein. We know the viruses has the DNA. But what we are concerned about is the bacteria cells. We're concerned with which one of our radioactive isotopes, sulfur or phosphorus, went into the bacterial cells. So what we want to do is we're going to look down here. And we want to see if we're going to see some red for the protein, because the sulfur was dyed red for the radioactive isotope, or are we going to see phosphorus for the DNA? And what they found here, guys, was at the bottom, when they determined what radioactive isotope it was, we had a very nice blue pellet. Okay? So what did this conclude? This concluded that our DNA was the thing that got taken over by the virus. So the virus injected the DNA into us, and that caused our DNA to change. All right. Now, at the time, they didn't know it was even our DNA that was getting manipulated. They just knew the virus was infecting us and injecting something into us, and they didn't know if it was the protein or the DNA. And this experiment determined that it was the DNA that was getting injected into us by the virus. And here's the pictures of all that, guys. All right, I just figured I'd draw it. I thought it'd be easier that way. Okay, so let's fast forward a little bit. This was still a little bit before Watson and Crick, but pretty much around the same time. We knew that DNA was made out of monomers called nucleotides, and we just went over that. We have a nitrogen base, whether it's adenine, guanine, cytosine, or thymine for DNA. If it is RNA, we add some uracil in there instead of thymine. It's a sugar. It has that. So DNA is deoxyribose. RNA is ribose. And it also contains a phosphate group. Now, they weren't sure how everything was put together. We knew the DNA had 2 strands, but we didn't know how those strands bonded together. Now, next guy, his name was Erwin Chargaff. He was able to determine the bonding of our nucleotides. So we have, this is DNA only, we have thymine, adenine, cytosine, and guanine here. Now, before I get over base pairing rules, I want to show you guys one thing. If you look at just the nitrogen bases, so we're just looking at the right side here, notice the amount of rings that are in these different nitrogen bases. Thymine only has 1 ring. Okay. Adenine has 2. So 1 here and 1 here. Like I said, thymine only has 1 ring. Cytosine only has 1. And guanine, 2 rings. Okay? So that's one difference, is that A and G have 2 rings. C and T only have 1. I got an easy way kind of for you to remember that A and G are together. So what Chargaff thought, actually I'm going to come back to this one, guys. Let me go back here. Originally, guys, they were like, okay, well, maybe it's A and A that go together, or maybe C and C bond together. We know it's not that anymore, but it's definitely not the same nitrogen base that bonds to another one. A and G. A and G. These are going to be called, they're nitrogen bases that are called purines. C and T, these are called our pyrimidines. Now, how I remember these guys, A and G are the purines, I always think of AP Bio. I don't know. AP Bio is pure agony. All right. Pure for purine, agony, starts with AG. A for adenine, G for guanine. Okay. That's how I remember it. And then how I remember that the amount of rings here is the purines, they have 2, but that's not how I remember it. I remember C and T, they're the pyrimidines. To me, that sounds like pyramid. Pyramid was always just one structure. You never had like 2 pyramids connected to one another, literally. That'd be kind of weird. It would kind of form like a trapezoid looking thing. But anyway, the pyrimidines, they all have 1 ring. The purines, they have 2 rings. So in order to keep it consistent on each side of our double helix, we are going to have 3 rings going from one side to the other side. So we're always going to have a purine bonded to a pyrimidine. let me show you what would happen if we did not do that. Say we bonded C with T. Okay? They both have one ring. And then we bonded A with G. They both have two rings, so they're going to be, a little bit wider. But our DNA would go in and out, like it would go like this. We don't want it to look like that. We want it to be a straight line going down each way that we can then twist. All right, so we want them to be parallel, the opposite sides to be parallel to one another. So what Chargaff found was that A always bonds to A always bonds to T, and G always bonds to C. And I kind of slipped with how I remember this, but it's apple in the tree and car in the garage. Okay, A always bonds to T, G always bonds to C. Apple is in the tree, car in the garage. All right, that's the easiest way I always think to remember that. Okay, guys, let's stop here before we go over Rosalind Franklin, and we will come back next time, and I will talk to you about the history of the discovery of that double helix. Have a good rest of your day, guys.